Hello friend, welcome to iBioca classes that deals with NEET, AIIMS, NISA, Olympiad. It was given by the teacher AKB sir, and you can visit all other details in other chapters at the site given below. Friend, today we are going to start the canal system of Porifera. In our last class, we have completed up to the body system, up to the body level of organization. So now the very important part of porifera is its canal system, which is that the sponge is organized in such a manner to form a complex system of pores and canals. This system is called canal system. That means the body of this sponge is normally it is having small pores present, and these pores are called as what? Ostia, and through this pore these are called as ostia, and through this pore what happens, these pores water enters through these pores, and so these pores form a special type of canal system within the body. Let's find what kind of canal system. So you can view this picture. So here the pores present, it is the outside part, and this is the inside part of the body, and in between the canal present in this part is the canal system, and through this part is otherwise we call them as is the canal, we call them as canal, and group of canal present we call them canal system. So friend, accordingly, in this picture, we got this canal system through which the water enters into the system, and the food is being absorbed by specialized cell, and carbon dioxide waste material is given to this particular cavity, and from this cavity it moves out through the body with the help of another opening called as osculum, and this middle cavity is called as the central cavity, and through whole body the spore canals are present. So together we call them as canal system. So accordingly, there are three basic type of canals present, and it is normally this canal system is meant for food gathering, respiration, and removal of waste materials. And normally in this diagram you see through this water enters and then water moves out. That means these are the canals, these are the special canals present, and through these canals together canals present in the body, we call them as what? A canal system. These are the canals together present, and the central cavity we call them as spongocoil. The first canal we find is a very simple canal called as the Ascon type canal. Let's know what are these Ascon type canals. In this Ascon type canals, normally we find there is no branching, and it is regarded as the simplest type of canal because and it is normally found in Leucosolenia, the organism Leucosolenia, and in few other organisms. Here what happens? Normally, here the what happens the canal present are very simple. That means the canals are very simple. They are not branched. They are just simple like this canals without any branching system. Having a dermal pore present ostia canal, and then directly they move into the cavity that is called as the spongocoil cavity. Next, move on the complex canals, little complex type that is called as the cycon type. And this cycon type is the more complex than the ascon type, and it is found in the species that is called as cycon, and some other sponges do also contains them. Here what happened friends, now here you find one branch here, another canal move, and here we find the branching system present. Here are the branching system, prosophiles, apophiles, then after branching they move into the cavity, and then they get exchanged, and whatever get exchanged then they get moved out from the body. But normally what happens, the canals are here branched, so little complex type, and it is normally found in cycon type. And the last one canal system that is called as the leucon type, it is the most complex type found in the species Spongilla, freshwater sponge Spongilla. Here what happens, the canal is so branched that it forms flagellated chambered like system is present. Here is one branch present, here branch present, here branch present, and these fragile chambers are attached to each other. So here dermal pores present, excurrent canals present, and here is the excurrent canals that gives out whatever materials has been given out or taken in. So likewise, it is regarded as the very most common having chambered form of canals. And and friend, we know. Let's move. what is the direction of movement in these canals? Normally, in Ascon type, the Ascon is very simple. As we know, they're very simple canals. So water simply moves through this, then moves, then into the cavity, and then to the osculum. So ingressing water, this is the ingressing water part, and the first opening present is the dermal part, dermal canals present. Then this part is the dermal canal part. Then we move on to the cavity. This cavity present is the spongicoil cavity, and moving out to the body, that is the osculum. So here what we find, that is very simple canal. Enter ingrown word. This is dermal canals. This part is the dermal canal part. Got it? This is the dermal canal part, and then we move on to spongicoil. Very simple. And next move the pathway of the second canal system, direction of movement, that is called as the cyclone type, which is a little complex type. What happens here? Let's move. What happens? Now in cyclone type, the ingressing water. Let's move. This is the ingressing water moves. They enter into the first canal, that is called as dermal canal. Spores present. Then inside they found, that means normally what happens? They enter. Once they enter into it, they get branched. They get branched. Okay, so these branches, these are the incurrent canals present, and then they move on to the special openings present called as the prosophiles present, and after prosophiles they again move on to form the other canals found, another branching form present, that these branching forms are called as the radial canals, and after radial canals the opening present at the end of the radial canals are called as apophyles, and these apophyles then directly open into the cavity, that is called as the spongicoil. So what happened here, we find the apophyles present. Here is the spongicoil, so it is the apophyles part, and this part is called as the spongicoil part, and in between small openings present, these are the small openings present in current canals opening present, and these are normally, these small openings are called as the prosopile parts. So this is little complex type normally found in cyclone. Then friend, let's move to the last canal system, and the last canal system direction of flow is quite simple, except they don't have radial canals, they have a flagellated chamber. So instead of radial, they have what? Flagellated chambers are found to be present. So these all are same to cyclone, only flagellated chamber in case of radial chamber. There we find radial chamber, here we don't find radial chamber. But overall, they're quite similar, but only they find specialized flagellated chambers are found to be present. Okay, friends. So this is the overall view. These are the apopiles present here, and these are the in between the radial canals present here, and the first canals are the incurrent canals, and prosophiles have with the openings present between incurrent and radial canals. The opening present between these two is called the prosophiles one present. So likewise, this is here we find the flagellated chambers present, and the canal that moves outside is called the excurrent canals present, and in between the flagellatum incurrent canals and ostium present, ostia is found to present. So this is the overview of the type of canals. Now friend moves to the function of the canal system. Normally the canal system helps in gathering of food. It helps in gathering of food materials, and the food materials are normally microscopic animals, organic particles, and this food are gathered with the help of this canal system, and they're absorbed through choanocyte cells, special cells present. I've taught in the last class, and these food get absorbed. Similarly, the exchange of gases takes place through the sponges through canal system as well as through the body layers. Then in Dermosphagia, especially sponge present Dermosphagia, the leucocyte type called the larval stage. In the larval stage, a special canal present that is called the ragone type. This ragone type is quite the spindle shape, both the end pointed, middle one is widened, and this type of canal is found in this ragone type, which that this central cavity opens to spongicle. Skeleton present, the skeleton are calcareous, that means they are made up of calcium carbonate, and siliceous spicules present, silica are found to be present with special type of protein fibers that is called as spongin fibers are found to present in the middle mesohyl layer. Next, friend, we move to its skeleton. The skeleton is quite complex types. Normally, the skeleton having six angles, six formed angle. They are called as hexactinellida. Some skeletals in Demospongia are modified, that is also made of silicus, and the fibers present, special collagen fibers present called spongin fibers, and these are the calcareous. They all are the spicules are calcareous. This is monaxon type. The three present triaxon type, and if four present tetraxon type, and if six present hexaxon types. So similarly, on the basis of angle, the canal has been classified. Let's move on to digestion. Digestion is normally present inside the body intracellular with the help of food vacuoles, as in protozoans. Similarly, sponges are microscopic food organelles. Choanocytes as the microvilli, they get absorbed and hence that means those choanocytes present having flagellated, so they absorb the food inside the food vacuoles. The food get digested inside the choanocytes and get stored in the layer, that is the mesohyl layer. It is get stored. And through this, now move on to the part that is circulation. Normally in circulation is also food is also brought about by the wandering amebocyte cells. I have already taught in the mesohyl cells. These are the layer. This is the inner layer. This is the outer layer. In between then special cells present in this mesohyl cells, and these are called as amoeboid cells. And these amoeboid cells helps in transporting circulation. That is, in between these layers, these are the amoeboid cells present. And this is the outermost layer, and this is the innermost layer. The innermost layer contains flagella, choanocytes, and spicules are present. All this, the middle layer is the mesohyl layer found to be present. That helps all these things. Now respiration, I have already taught gases exchange through simple diffusion through the plasma membrane or through like protogens. Excretion, which is the metabolic waste product, also occur by diffusion through plasma membrane. Chief excretory material is ammonia. So ammonia is the chief excretory material. So all of them, so through simple diffusion, they enter into it by simple diffusion. Now friend, let's move on to its last part, that is reproduction sponges. Normally, sponges reproduce both asexually and sexually. Reproduction occurs in sponges asexually by fragmentation. That means sponges get into small fragments, and each fragments then develops into what? Into a new individuals or new each sponge develops into new individuals. So these are called as fragmentation. Sometimes asexual reproduction also takes place with the help of budding, and special internal budding presence called as gametes. Budding means sponge gives out external outgrowths present outside the cell, that is called external budding. And if sponge gives internal buds, these are called as gamules. And these internal buddings which are present, the gamules. The gamules contain specialized cells present, and these specialized cells present inside these are the specialized archaeocyte cells present. And so normally gamules are the internal buddings present, and they contain specialized archaeocyte cells present. And these archaeocyte cells are these all cells are the archaeocyte cells present, and it is externally covered as well as internally covered. In between them, the spicules are present. Okay. So in adverse condition, what happen? Through this opening, micropyle present opening, and through this, these gets released out, and they release out and then gives out into new individuals. So these are normally regarded as the internal budding type of sponges. Now, at last, sexual reproduction. Normally, sexual reproduction is hermaphrodite, that is monoecious species. That is hermaphrodite. Power of regeneration is maximum. And this in sexual reproduction, what happen? Now the fertilization is internal. That means one of the this is sponge A, this is sponge B. One sponge, two sponge. The sperm transport to this sponge or from this sponge. As a result, transport do takes place from one sponge to another sponge, and inside the body, the fertilization results. So it is called as internal fertilization. So N plus N, they form 2N. That is called as zygote. So zygote is produced by this internal fertilizations. So sperms of one leaves and enters another with the water current to fertilize the ova egg. Instead of fertilization internal, it results in diploid zygotes normally. Then development, normally we go know that the zygotes to produce, the zygote does produce is holoblastic. That means complete division. That means total zygote cleavage is totally divided. Total division of zygote do takes place, and the development is indirect. Indirect, that means the development, that is the young stage, is present in between the adult stage. So in between young and adult, another stage is present. Is free swimming. That let's move on to development. In this development, we should know that the zygotes so formed, the zygote of porifera are holoblastic cleavage. The development that means complete division. Suppose this is zygote, it completely divides. It completely divides. But in meroblastic, what happens? Partial part of it gets divided. In meroblastic, not in this. Let's move. So it has been estimated that the developments of form is of larval type, because we find, suppose this is zygote, the zygote develops to produce the larva, and this larva then develops to give rise to the adult. So what happens? Now this zygote divide, so it is having it, a stage in between the adult and the young, that's a larval stage. And this larva is a free swimming larva. It is called as amphiblastula in Sycon, whereas it is named as parenchyma in Leucosolenia, the dispersal of species. So development is always indirect. If larva is produced in any of the cycle, then it is called as indirect development. Now, what are the unique features of this phylum? That is, first of all, that it contains ostia and osculum. Second is that the cellular level of body organization. That means they have got pinacocyte cells and choanocyte cells having flagellated. So these cells and archaeocyte cells present. These three basic type of group of cells helps in amoeboid cells in in its level of organization. Then we find a special canal system present, and we got that they do not have any digestive cavity present, but a spongicoil cavity is found to be present. No digestive cavity is present. Then we found that presence of choanocyte cells, and these choanocyte cells are the specialized flagellated cells having, and they help in absorbing food materials and having vacuoles for internal digestion of foods, and they have got skeletons present in the form of spicules and spongin fibers. So, and what are the advantage over the protozoa? First of all, protozoa is a unicellular, and this is a multicellular larger body, and we find that here division of labor present because there are different type of cells. So coordination between the cells do takes place. So divisional level is found, and it contains special sperm present, egg present, means gametes are found to present. These special character gametes present, and the unicellular zygote undergoes cell division to produce embryo, but we don't find embryo stages in protozoa, but it is found in, that is, porifera. Now move on to classification. On a modern basis of classification, there are four types on the basis, and this classification is based on the mode of skeleton. One is hexactinellida, one is calcarea, another is demospongii, another one is sclerospongii. Let's move about the classification. And the first classification is called as the calcareous type, and this calcareous type, it is called because the skeleton is made up of calcium carbonate, and the spicules are of monoxone type, trioxone type, or tetraoxone type. That means either they are very simple, or they are of three basic type, monoxone type, or they are of four basic angles present, trioxone type. It is mostly present Leucosolenia, Sycon, and Grantia. Next, classification, that is called as demospongians type. In demospongians type, hexactinellida type. In hexactinellida type, the hexagonal structure is found to be present, and hexagonal structure means they're present of six type present, six angles from siliceous spicules, 1, 2, 3, 4, 5. The six group of rays are found to be present, and they are silicon based spicules, and is normally found in Euplectella, otherwise called as Venus flower basket, or Hyalonema, that is also called as glass rope, special named present. And the last or the foremost, we call them as the demospongia type, and the demospongia type, a dermal type of found, that is, it is made up of spongin fibers, is found to be made of fibers, may contain spongin fibers, which is of silicon types. Skeleton may be absent, but the skeletons, if present, they are of monoxone type or tetraxone type, but maybe eight red stays present. but no six-tailed stage is not found to be present. Got it? And it is mostly found in Euspongia, otherwise called as the bath sponge, or it is found in Spongilla, otherwise called as the freshwater sponge, or it is found in Cleonia, called as boring sponge. This boring sponge, or Chalenia, we call them dead man's finger, or mermaid's glossus presence. Got it? So these are the basic classifications we have come to know. And friend, this is all about your porifera. And in the next video, we'll deal with Nidaria. And hope you enjoyed my video, and hope you go through my next video or the next phylum, that is Nidaria. And more details you'll get in our site, website, that is www.iibiocare.com. Okay, thank you.